Okay, so welcome to this next video in which we are discussing palmitoylation of proteins and palmitoylation inhibitors. Okay, so we were in the process of discussing the DHHC family of enzymes, which contains 23 members which are involved in catalyzing the S-palmitoylation of proteins. So basically, the exchange of the palmitoyl group from the thiol group of coenzyme A onto the thiol group of cysteine residues in the target protein is not something which occurs just spontaneously. It's something which requires an enzyme to catalyze, and these DHHC enzymes catalyze that. Okay, so we discussed that they are integral membrane proteins which are anchored in lipid bilayers, and we're in the process of discussing which lipid bilayers each of the 23 forms of DHHC enzyme are located in. So we've discussed those DHHC enzymes which are strictly located in the ER, and we've discussed those that are located in both the ER and the Golgi. We now want to discuss those that are strictly located within the Golgi. Okay. So, these DHHC enzymes that are located in both the, located strictly in the Golgi include DHHC3, okay, in orange here, DHHC4 in orange here, also DHHC7 and DHHC8 are also located specifically in the Golgi apparatus membrane. And then we also have DHHC15, 17 and 18. So 17 is one of these ones down here, whoops. And then DHHC 18 over here is also located specifically in the Golgi membrane. Okay, now let's go on to the plasma membrane. So which DHHC enzymes are only located within the plasma membrane? Well, this is DHHC5 in pink, DHHC20 and DHHC21. So those three are all located in specifically the plasma membrane. And now finally, to culminate, we'll look at those that are located in both the plasma membrane and the ER membrane. And the two which are located in both the plasma membrane and the ER membrane are DHHC23 and DHHC24, the remaining ones. Okay, so we've now discussed the structure of the 23 different forms of DHHC enzyme, and we've also discussed which membranes within the cell they are localized to. Okay, now I just want to discuss the DHHC region of 4 amino acids in a bit more detail, because this region is present in absolutely every single DHHC enzyme. Okay, this conserved sequence of 4 amino acids, and the reason it's conserved is because it's extremely important in catalyzing the transfer of the palmitoyl group from the thiol group of the coenzyme A molecule onto the thiol group of a cysteine residue within a protein. Okay, and basically what happens is that the palmitoyl group from the palmitoyl coenzyme A temporarily goes onto the cysteine residue within the DHHC region of the DHHC enzyme, and then it is transferred from the thiol group of the cysteine residue of the DHHC sequence in the DHHC enzyme onto a cysteine residue within the target protein. Okay, so basically the palmitoyl group firstly goes onto the enzyme, onto a cysteine residue within the enzyme, and then is transferred onto the cysteine residue within the protein. And it is specifically this cysteine residue here within the DHHC sequence of 4 amino acids that receives that palmitoyl group. Okay, right. So that now concludes our discussion of palmitoylation. Let's now turn our attention to palmitoylation inhibitors. Now there is one fantastic palmitoylation inhibitor, okay, and this is called 2-bromo-palmitic acid. So we'll begin by discussing what the structure of 2-bromo-palmitic acid is. Okay, so basically it's palmitic acid with a bromine atom, which is a halogen, attached to the second carbon. Okay, so let's go again. So remember, palmitic acid is a 16 carbon fully saturated carboxylic acid. Here's carbon 1, here's carbon 2. We now need a bromine atom coming off carbon 2 and also a hydrogen. And now we need the other 14 carbons. So 13 of these are going to be a methylene group. So here's 1. Okay, and I'll just put a bracket around this to subscript it with 13. Okay, so that's now the 13 methylene groups, and then at the end you'll have a methyl group here. Okay, so if we tot up the number of carbons now, we've got 14 here and 2 here. That takes us up to 16. Okay, so this is 2-bromo-palmitic acid, and for short, 2-bromo-palmitic acid is abbreviated to 2BP. Okay, for bromo and then palmitic acid. Right, okay, now this is a stable molecule within cells. If you put this into cells, it does not get broken down, it does not get metabolized. And the reason it doesn't get metabolized like other fatty acids like this would be metabolized is because of this bromine on position 2. This prevents beta oxidation, basically, which, remember, breaks fatty acids down into acetyl molecules. Okay, so this bromine here is going to stop that process, and therefore the 2-bromo-palmitic acid is stable within cells. Now, as soon as you put it into cells, what happens is that it joins on with coenzyme A molecules, just like the normal palmitic acid would, and it forms 2-bromo-palmitoyl-CoA, okay, which is often abbreviated to 2BP-CoA. So, and this is short for 2-bromo-palmitoyl. So I'll write it out in full. So the B is for bromo, then the P then is for palmitoyl, and then CoA, which obviously is short for coenzyme A. So, when you put it into cells, it will combine with the coenzyme A to form a thioester link in just the same way that the palmitic acid would form a thioester link with the thiol group of coenzyme A, and forms this molecule called 2-bromo-palmitoyl coenzyme A. Now, the mechanism of action of 2-bromo-palmitic acid is not known, although I will I will give the 3 sort of mechanisms which people believe may be how this works, but it is an extremely effective inhibitor of palmitoylation. Okay, so if you expose cells to 2-bromo-palmitic acid, they stop adding palmitoyl groups onto their proteins, basically. So it's extremely potent palmitoylation inhibitor. Okay, but the mechanism isn't fully understood, although I will give some suggestions for how it could work in a moment. Now, what's interesting is that close analogues of 2-bromo-palmitic acid are not effective at inhibiting palmitoylation. So, for instance, if you create 2-hydroxy-palmitic acid, which is where you put a alcohol group off the second carbon rather than a bromine atom off that second carbon, this is not effective at all at blocking palmitoylation. Another example is if you have 16-hydroxy-palmitic acid, which is where you put an alcohol group off the 16th carbon here. Okay, that's not an effective inhibitor of palmitoylation at all either. Another important example is if you use palmitoleic acid. Okay, so palmitoleic acid. This doesn't inhibit palmitoylation either. Okay, and we'll see how some of these not inhibiting palmitoylation may sort of swing your opinion on which of the 3 mechanisms is likely to be the mechanism by which 2-bromo-palmitic acid works. Now let me just show you the structure of palmitoleic acid. Okay, so again it's a 16-carbon carboxylic acid, but this time it's got a double bond basically, and this double bond is going to be between the 9th and the 10th carbon of the carboxylic acid molecule. Okay, so we can go up to carbon 8 then, so I'll put a methylene group here, and then if I put 7 there, then that takes us up to the 8th carbon. Then here is the 9th carbon. We want a double bond between the 9th and the 10th carbon, so put 1 hydrogen off each of these carbons, and then, whoops, what am I doing here? We want the methylene groups coming off here now, so that's the 10th carbon there. So how many methylene groups are still to go? Well, 5 are still to go, and then the final carbon will be a methyl group. So here's the 5, and then here's the 6th one on the methyl group right on the end. Okay, so that takes us up to 16 carbons. We've got 9 over here, then we've got 10, and then another 6 here. So that's palmitoleic acid. Okay, so this isn't an effective inhibitor of palmitoylation either. Okay, so let me talk about the potential mechanisms by which 2-bromopalmitic acid works. So, the sort of leading thought for how 2-bromopalmitic acid works, the sort of hope, what everyone hopes is the mechanism by which 2-bromopalmitic acid works, is that it inhibits the DHHC enzymes, basically. So, inhibition of the DHHC enzymes. Right, so how do we think it might do this? Well, remember me telling you about the DHHC sequence within the cysteine-rich domain, and remember me telling you about how when the DHHC enzyme is catalyzing the palmitoylation of a cysteine residue within a protein, what first happens is that the palmitoyl group is exchanged off the coenzyme A onto the cysteine of the DHHC sequence. And then this palmitoyl group is exchanged off that cysteine onto the cysteine of the target protein. Well, the thoughts are that maybe 2-bromopalmitic acid adds on to the cysteine of the DHHC sequence within the DHHC enzyme. So effectively, if I draw a little picture, and I'll use the most common DHHC form, okay, so this form where you have these 4 membrane spanning alpha helices like so, okay, and this is the cytoplasmic side here, and this is either the extracellular fluid or the luminal side, and basically we'll have a cysteine somewhere in here which will be the cysteine of the DHHC, and what will happen is the 2-bromopalmitic acid will add on to there just like it's supposed to, just like a normal palmitic acid molecule, but then the thoughts are that it, unlike the palmitic acid molecules which are then transferred on, it can't be transferred on, so it gets stuck on there, and once it's stuck on there, then there's no way this enzyme can catalyze the palmitoylation anymore, and therefore you have inhibited the enzyme. Okay, so that is kind of the mechanism that everyone is hoping is the mechanism for 2-bromopalmitic acid. Okay, so let me discuss some other theories, however. Other theories are that it actually is put onto the proteins. So, basically, what will happen is the 2-bromopalmitic acid will go onto the cysteine of the DHHC sequence of the DHHC enzyme, and then it will be transferred onto the protein. So let's say this is our protein target. Then you do end up putting it onto the protein. The thing is that once you've put this 2-bromopalmitic acid molecule onto the protein, it doesn't do what a normal palmitic acid molecule would do. At least this is the second theorem. So the first theorem was here. Now let's discuss this second theorem here. Okay, the second theorem is that you do add the 2-bromopalmitic acid molecules onto proteins, but they just don't act like normal palmitoylation. Okay, so effectively what you do is when you put the huge amount of 2-bromopalmitic acid into the cell, all of the palmitoylation occurs with 2-bromopalmitic acid rather than normal palmitic acid. So all of your proteins get 2-bromopalmitic acid added onto them instead. Okay, and basically the idea is that this will not anchor in phospholipid bilayers because of the bromine atom, which means that this bond is polar here, and therefore suddenly you've got a less hydrophobic region here, a more hydrophilic region, which won't interact so favourably with the hydrophobic core of the phospholipid bilayer. Okay, so the idea is that you do still add the 2BP molecules on, but that they don't function at all like they should, okay? And they block normal palmitoyl groups being added onto the proteins, and that is how they block palmitoylation. Okay, so they're effectively a competitive inhibitor for the palmitic acid molecules. They're put on instead, and they don't do what the normal palmitic acid molecules would do. Okay, so what should I title that? I'll just put competitive addition with palmitic acid molecules, maybe. Competitor for palmitic acid, okay, which I'll just abbreviate to PA. Right, and then the final theorem is that potentially what could happen is the 2-bromopalmitic acid molecules, which are known to inhibit loads of other enzymes in the cell, and all of these other enzymes are involved in lipid metabolism. So the other idea is that basically by inhibiting certain enzymes that are involved in lipid metabolism, what can happen is it could stop the production of normal palmitic acid. So stop synthesis of normal palmitic acid is the third concept that could be the mechanism for 2-bromopalmitic acid. Stops synthesis of palmitic acid. Okay, so if it stopped the synthesis of normal palmitic acid, then obviously if you've got no normal palmitic acid, then you can't add it onto proteins, and therefore you stop palmitoylation. Okay, so all of these are potential mechanisms by which 2-bromopalmitic acid works to prevent the palmitoylation of proteins, and as of yet, we do not know which of these is the mechanism by which 2-bromopalmitic acid stops palmitoylation.